today we will discuss about the cancer role and metabolism. So how it relates about the health disorders. So that is the major aims of cancer. So cancer cells is they are characterized by three properties. They are like a diminished and unrestrained control growth. So invasion local tissues and spread like metastasis to other parts of the body. So nearly all cancer cells, like cancers, originated from the single cell of this clonal origin is typically descriptions of features like between the neoplasia and hyperplasia. So physiological, like physical, chemical, biological agents like can cause to the cancers, like physiological, chemical, biological, some agents, so these factors they cause cancer. So agents causing cancer fall into the three broad categories like radiant energy, chemical compounds, and viruses, spontaneously mutations, and oxidative damage to DNA. So some of the relations about carcinogens interact with the covalent with the cellular macrocell molecules. Carcinogens found to interact with the purine terminals and the phosphodiester groups of DNA. So the most common site attack guanine, and in addition various carcinogens like N2, N3, like nitrogens 1, 2, like 2, 3, and oxygen 6, 8 atoms of the base, like these conditions. So then cancer is major role is persistent in unpaired lesions are important generating mutations of critically for carcinogenesis. Like ambassador like is used to detect the mutagenicity of the chemical carcinogens. Like MES like the uses especially like constructed strains and salmonella and typhimurium that has mutations in gene that codes for one of the enzymes involved in synthesis of histidine. So like salmonella typhimurium, like the above the strains can be not synthesized histidine, needs to histidine added to the medium for its growth. Chemical carcinogen by its mutagens natural restore synthesis histidine the typhimurium. Salmonella typhimurium along with the mitochondrial supplementers provide the activations by monooxygenases. Initiation and promotion. Coming to the initiation and promotion, so in organs such as the skin and liver, carcinogens like carcinogens has two stages: initiation and promotion. So initiation is rapid, irreversible stage, rapid and irreversible stage. Most carcinogens are capable of acting of both initiating and promoting agents. So DNA is the critical macromolecules in carcinogenesis. So both radiations and chemical carcinogens damage and DNA are capable of causing mutations in DNA. Many tumor cells exhibits the abnormal chromosomes. So some transfections experiments indicate that the purified DNA oncogenes form cancer cells can transfer the normal cells into the potential cancer cells. Genes that increase the susceptibility to cancer have been isolated. Epigenetics is identified as changes that alter pattern gene expressions that persist across at least one cell deviation, methylation like dinucleotides and histone acetylations. Some of the relations about the cancer cells and some changes shown that culture suggested that malignant transport has occurred insufficient and oncogenic viruses. However, the crucial test malignancy is the ability of cells to grow in vivo. So alternative morphology, transformed cells often have like much rounder shape that control cells. So increased cell density, loss of contact inhibition growth and transformed cells from the utilities, while the control cells usually from the monolayer. Loss of encouraged independence, the transformed cells grow without attachments to surface to culture dish and it will grow in some agar. So loss of contact, loss of contact inhibitions of movement, the transformer cells grow over one another, while normal cells stopping the moving to when they come into the contact with each other. So a variety of biochemical changes, including an increased rate of glycolysis. Alternatively, the cell surface, the changes in the compositions of glycoprotein, glycolipids inside and secretion certain proteases. Alternatively, cytoskeletal structure such as actin filaments and dimension requirements for growth factors, and often it increases the secretions of certain growth factors into the surrounding medium. So, oncogenic plays crucial roles of in carcinogenesis, and oncogen is a gene when mutated or expressed at high levels turn normal into the tumor cells. Oncogens were first recognized as a unique genes tumor causes viruses that are responsible for the process of transformations. Some of the oncogens are Ross sarcoma viruses. The Ross sarcoma viruses is retroviruses containing 4 genes named as GAG, POL, and SARC. These are the genes. The GAG genes encodes with the group of specific antigens of the viruses, and POL gene for the reverse transcriptase that characterize retroviruses, and for certain glycoprotein viruses, sarcoma causing gene produce a protein tyrosine kinases. These abnormal phosphorylations of insulin a protein fecal adhesion plateaus could be help explain the rounding of the cells, their adhesion of susceptible to one and another during the transformations. Certain glycolytic enzyme appear to be target proteins for SARC tyrosine kinases. So the producers of SARC interact with the kinases, catalyzing phosphorylations of phosphatidylinositol and phosphatidylinositol 4,3 and 4,5 biphosphate. Inositol triphosphate releases the calcium form intracellular storage. So diacylglycerol activate the protein kinase C, which in turn the phosphorylates number of proteins, some of which are in ion forms. Mild alkalizations of the cell brought about the activations of sodium hydrogen antipode system could be play a role in stimulating mitosis. So protein kinase, protein tyrosine kinase in the normal and transformed cells insulin and platelet derived growth factor and end derived growth factor found both normal and transformed cells tyrosine kinase activity. The amount of phosphotyrosine in the most normal cells is low, but is usually elevated in the cells transformed by the oncogen viruses containing protein tyrosine kinases. So how it regulates means the product RAS oncogen is a Moran sarcoma viruses binds with the GTP, and as GTP is as activity regulates the activity of adenyl cyclase, and oncogen encodes the DNA binding proteins. Albeit cis oncogen products have tyrosine kinase activity. Some proto-oncogens that gene can be that proto-oncogen gene can be oncogen due to mutation or increased expression. Proto-oncogens believed to play important role in normal differentiations differentiation and other cellular process. So oncogen cells, tumor cells, DNA isolated from the tumor cells. The recipient added recipient cells often hormone mouse fibrinolysis and microscopic observation 4 to 2 weeks, and the foci of transformed cells. So the procedure while talking about the procedure is repeated several times of using DNA extraction from the transformed cells. Those reducing amount of DNA not only involved in transformation that was transferred and facilitating identification by the southern blot technique using suitable probe. So activation of proto-oncogen, the 5 mechanisms of activations of proto-oncogens: the promoter insertions, enhancement insertion, chromosomal translocation, gene amplification, and point mutation. These are the 5 proto-oncogens mechanisms this activation of proto-oncogens. So proto-oncogens insertion certain retrovirus lack of oncogens avian leukemia viruses, but maybe also cause cancer over a long time period. So retrovirus infect the cell's DNA copies and complex DNA. Their RNA genome is synthesized by reverse transcriptase. And also the complex DNA is integrated into the host genome. So how it regulates means these are the mechanisms of actions of oncogens. The oncogen acts as an intracellular pathway involved in the growth control, uncoupling them from the need for exogenous stimulus. Products are acting as a thyrokinase product of RAS, acting to stimulate adenyl cyclase, act as a phosphorylating key regulatory protein of cell cycles. So how it will regulate means some of the conditions like elevated various cyclin decreases the kinase inhibitory and KIB, P31, P21, P25, P27, P57 decrease the inhibitory of CDK4 and INK4, P16 has been documented in cancer. So growth factors, the polypeptide growth factors like are mitogenetic and like mitogenic growth factors act in endocrine, paracrine like autocrine manner. Growth factor act on the cell cycle mitosis via the transmembrane signal transduction. So growth factors and oncogenes interact in the several ways. So the product of several oncogenes are either growth factor or parts of the receptors for growth factor. So product of cis oncogene transduced like beta chain blood derived growth factor. Beta chain is biological active homodimer without the involvement of alpha chain. So the autocrine stimulations of by prolactin derived growth factor gives and chronic mitogenetic stimulus would be an important factor in the transformations of cells. The product of ERP like a turn cated and like NLEC growth factor, much of deletions of external domain and but retains the tyrosine kinase part. This abnormal form of continuously active tyrosine kinase when present in the cells of causing chronic mitogenic stimuli. So how it is transforming means growth factor beta is transforming growth factor beta known as inhibiting growth factor. Most of the cells types of express fibroblast. This fibroblast that transforming growth factor beta promotes the growth factor activating gene. This transforming growth factor beta gene inhibitory effect is on the cell cycle progression and phosphorylating reduction of levels of mRNA, cycling and E and A, inhibition the cycling and A, depletion the kinases. How it will be the mechanism and some of the properties of RP1 gene means and the protein product. So gene is located in on the 13q gene. So that the familial retinoblastomas have identical mutations of both allele loss. So this unphosphorylated species of P or B gene certain viral proteins like large T antigen the forming of complexes of the activations of the. So mutations in the in these genes are also involved in the osteosarcomas and certain other human tumors. So the P53 tumor suppressor gene act as an guardian of the genome. So mutations in the P53 occurs frequently in many human tumors. So these are the some of the P53 gene and it is protein product and gene is located into the short arm chain like chromosome like 17 part this product is P53 is nuclear and phosphoprotein 53 KDA with 5 major structural region P53 binds with the specific sequence. Some also P53 binds with the variable some of the major viruses, various viruses, proteins. Example SV4 large and T antigen forming the inactive oligomeric complex. P53 is required in the normal cells and developmental, but absorption sequence of knockout mice and greatly increases the occurrence of tumors and associated with gene instability. So some selenium has cancer preventive action. Here we are talking about one of the micronutrient that means trace element that selenium has cancer preventive action. Selenomethionine, the main form of the selenium in our diets participate in the redox reaction resulting reduction in the two cysteine residues within the P53 leading to the index inducing P53 DNA binding the activity. HPOY is proteins E6 and E7 binds with the inactive cellular tumor suppressor P53 and PRP respectively. Some of the major features of P53 mutations will found out the human tumors. So later, like that, mutations in the p53 gene, the most common genetic alternative human cancer, and frequently colon, like breast and lung cancers. So these are the things we will discuss about the cancer and cancer related about the topics, and the mutations also some of the found with the highly conserved the codons. So these are the conditions we explain the cancer role, and the topic and related about the genes and oncogenes, proto-oncogenes, how related about the metabolism, so how it will be regulations in the further conditions, so that further condition will help. So that thing will conclude at this point. Thank you.